My name is Bobby Gaspar. I'm one of the founders and CEO of Orchard Therapeutics, and it's a pleasure today to talk to you about Orchard and the work that we're doing. So, Orchard is a global leader in hematopoietic stem cell gene therapy, and you can see from these pictures that we are a clinical stage company. The work that we have been doing in the use of HSC gene therapy has had the ability to treat a number of rare inherited disorders that I will talk about. And we believe we are making a real difference to these devastating diseases through the use of HSC gene therapy. These are my disclaimers. And so the first thing I want to do is really talk about the HSC gene therapy platform approach. And it's important to just talk through this because we believe that HSC gene therapy offers a highly differentiated approach. Not all gene therapy is the same. What we are doing is taking hematopoietic stem cells and introducing a working copy of the missing gene into those hematopoietic stem cells so that they can then potentially correct a whole series of different diseases and also have a durable effect. So I'm going to talk you through how that happens and about the potential of these genetically modified hematopoietic stem cells. So HSCs are blood stem cells that are found in the bone marrow. They have the ability, as we see here, to differentiate into multiple different cell types. So they can become immune cells, they can become red blood cells, they can also become a specialist cell type that can cross the blood-brain barrier and enter the brain. The other really important property of HSCs is the fact they have the ability to self-renew. So these HSCs renew themselves as well as giving rise to all of these different cell types that I talked about. To introduce a working copy of the gene into these HSCs, we are using lentiviral vectors. The lentiviral vectors enter the cell, and they integrate a working copy of the gene into the genome of the target HSC. Now, after that integration, once the HSC self-renews or whether it divides into a more differentiated cell type, because the gene is integrated into the genome, with every cell division, that genetic information is passed on to the daughter cell. And so this is why when we give gene-modified HSCs to a patient, through self-renewal and through the integration of that gene into the genome, we get a long-lasting and durable effect. In fact, a single administration of gene-modified HSCs has the potential to be curative. We have already started to see that potential and the durability in our programs. So in our clinical programs of gene therapy using HSC gene therapy, we've treated over 170 patients. In fact, the first patients in our licensed program, in our approved program for Strimvelis, were treated over 19 years ago. Using lentiviral vectors, the first patients were treated over 9 years ago. And so you can see that there is this large body of data with extended follow-up, which demonstrates both the efficacy and the potential safety of this HSC gene therapy. And again, I would emphasize all of this is after a single administration of gene-modified cells. One area that we have been able to tackle using HSC gene therapy is in certain neurometabolic disorders. And I'm going to give you an overview of some of the work that we've been doing in some devastating conditions. Metachromatic leukodystrophy is a really severe, rapidly progressive neurodegenerative disorder. A single gene defect leads to the accumulation of certain substrates in different parts of the body, but especially in the brain, in neurons, that causes neurodegeneration. In this sad story that we see here, here's a little girl who pre-diagnosis at the age of 5 is living a normal life. But once this disease kicks in, it really has a devastating effect. And at the age of 9, she's unfortunately bedridden, and her life expectancy will be significantly curtailed. At the moment, there is no therapy available for MLD, and the only option, which is a bone marrow transplant, has variable and inconsistent effects. For many children, palliative care is unfortunately the only option. In our program of HSC gene therapy for MLD, we have seen some potentially remarkable effects. I'm showing here on the left-hand side the improvement in motor function as a result of using gene therapy. The disease can be split into an infantile and a juvenile form. This is a score called the growth function score, which goes from 0 to 100. And this is the score that you see in untreated natural history patients. And you can see they have very low scores because they are so badly affected and unable to move independently. Children who have been treated with our HSC gene therapy have significantly improved motor function as a result of that once-off administration. And this is 3 years after treatment. You can see here the cognitive function in children treated with HSC gene therapy. And again, this is the cognitive age equivalent score. This is the chronological age. And very simply, children of 3 years of age should have a cognitive age equivalent score of 3. This dotted line represents the normal trajectory, and this gray shaded area, the range. These are the children in light green who have not been treated. They have very, very low scores because of the severe effect of this disease on their learning and cognitive ability. But children who have been treated with HSC gene therapy show a cognitive age development that is in keeping with normal children. And again, as a result of a once-off administration of gene-modified cells, they retain the ability to learn and interact with the outside world. We've taken this HSC gene therapy to other conditions as well. This is another neurometabolic condition called MPS1 or Herlocentrin, which affects, again, cognitive function. It also affects the skeleton as well, and children have poor growth ability as a result of this disease. What we've been able to show is that we, after infusing gene-modified cells into these patients, these cells engraft and are present in the periphery up to now 18 months after gene therapy. The substrates that accumulate in MPS1 now come down to normal levels, and this is both this substrate called dermatid sulfate and also heparin sulfate as well. What does that mean as far as clinical outcomes are concerned? Well, we are now starting to see early clinical evidence of protection from neurodegeneration in these patients. So these are the first 2 patients who have gone beyond the 12-month time period after which we can do these clinical assessments. And this is the first patient. And again, what we're seeing here is the development age, the chronological age. This is the trajectory in a normal individual. And this first patient here, who started off with an IQ of approximately 85, has continued to have a similar IQ as he has grown older after gene therapy. So there has been no deterioration so far. Similarly, this second child, who started off with an IQ of 100, continues to have an IQ of 100 12 months after gene therapy. These are early encouraging signs that gene therapy is protecting them from a loss of cognitive function. So we are building upon our strength in neurometabolic diseases. We have our program in MLD has been filed with the EMA. We have proof of concept studies in MPS1, as I've just shown you, but also we've started a study in another condition called MPS IIIA or Sanfilippo syndrome. And we also have a preclinical program in Sanfilippo B or MPS IIIB. In addition to that, because of what we're seeing in these ultra-rare diseases, we are looking to expand the use of HSC gene therapy into more common indications as well. And we've talked recently about a research program in a genetic subset of frontotemporal dementia. And there are other developments in larger indications that we are now collaborating with an expert in the field, Dr. Alessandra Biffi. And so I'm just going to talk now about our extension into less rare conditions. And one of the reasons why we want to move to less rare conditions is because we have been able, through our work in ultra-rare conditions to be able to understand the mechanism by which HSC gene therapy can make a difference in the brain. We've been able to understand that a subpopulation of genetically modified HSCs has the ability to cross the blood-brain barrier. When it enters into the brain, these cells differentiate and distribute throughout the brain. They differentiate into microglia, and because they are now gene modified, they are expressing the missing enzyme or protein, which can then be taken up by neurons and prevent the accumulation of substrates and prevent neurodeterioration. And so, having understood how that works in some ultra-rare conditions, we want to use that information to translate into larger indications. And so, we are looking at a genetic subset of frontotemporal dementia, and we'll be talking about the specific genetic subset and the patient population later this year. Similarly, we have been looking at how HSC gene therapy can also affect other tissues as well. And one important tissue is the gut. So in a rare immune deficiency called chronic granulomatous disease, which has very severe colitis, we've seen that HSC gene therapy can actually prevent that colitis and actually improve the colitis in patients who have very severe forms. And the reason for that is because in these diseases, a subset of cells in the gut called tissue-resident macrophages are defective. When there is bacterial invasion, they can't repel and control that bacteria, and therefore they set up an abnormal immune response, which causes the colitis. Through HSC gene therapy, we can now have migration of cells into the gut that differentiate into tissue-resident macrophages, which now can sense bacteria and mount the appropriate immune response and prevent that colitis. So having understood and seen this working in a rare form of colitis, this chronic granulomatous disease, we now want to apply that to a larger form of colitis, a less rare form of colitis, and we have identified a genetic subset of Crohn's disease where we think this may be applicable. And again, we'll be talking about this later this year. The other point I want to touch upon is our manufacturing strategy. We are looking at how we can improve the manufacturing of HSC gene therapy products through better transduction of the hematopoietic stem cells by better ways of making lentiviral vectors. And we recently licensed some technology to make a stable cell line that will make lentiviral vector more robustly and at scale. We'll also look at a processing of the transduction of these cells in closed and automated systems. Initially, we will partner with our CDMOs and build our CDMO network. And in the future, we will look to build our own in-house manufacturing capacity. And so over the last 6 months, we've made some real advances in supporting our new strategic plan. We've prioritized our portfolio investments with an emphasis on neurometabolic disorders. For example, we've highlighted the work that we've done in MPS1 or Herlocentrin, some of which I've talked to you about today. We've started our program in MPS 3A, and now 3 patients have been enrolled into that program. We've invested in our next generation manufacturing, and one piece of that was to license in the technology for the stable cell line. And we've also extended our manufacturing relationship with MolMed through to June 2025, so increasing our CDMO partnership. We believe that HSC gene therapy has the potential to really make a difference, not just in ultra-rare conditions, but also in larger indications as well. And so what we want to do at Orchard is to realize the full potential of these HSC gene therapies. We're investing in high-need, high-value programs, and also in early R&D to bring larger indications to clinical application. And we are building a sustainable and successful commercial infrastructure through our investment in manufacturing, building up our manufacturing network, and also building out our commercial infrastructure for the commercialization of these programs. Thank you very much.